This demonstration illustrates how an inhibitor can affect a catalyst in terms of causing a reaction to occur. In these two test tubes, we have hydrogen peroxide. Hydrogen peroxide naturally breaks down to form oxygen gas and water. However, the decomposition of hydrogen peroxide is very slow in the presence of light, which is why it looks like nothing is occurring in our test tubes. So we're going to add a catalyst, specifically iron 3 nitrate. The iron metal acts as a catalyst in the decomposition of the hydrogen peroxide. You may be able to see that there's some bubbling that is occurring, and this is due to the breakdown of the hydrogen peroxide. So the reaction rate is dramatically increasing. You can start to see that the reaction is becoming more vigorous as it's starting to actually move up the test tube. And you'll start to see that there's a small amount of bubbling continuing in the test tube due to the breakdown of the hydrogen peroxide. However, it is relatively stable now due to the reaction being essentially complete. We're going to perform the same experiment in the other test tube. However, this time we're going to end up adding an inhibitor. So again, we can see that the hydrogen peroxide is breaking down. We start to see this because there is the bubble formation indicating the formation of oxygen gas due to the decomposition of the hydrogen peroxide. I just added in sodium phosphate. The sodium phosphate is an inhibitor, which you can see has slowed down the reaction rate or inhibited the process of the breakdown of hydrogen peroxide. You'll notice that there's still a small amount of reaction occurring, and inhibitors do not completely stop a reaction, but instead they often slow down the reaction by binding to the catalyst. In this case, the sodium phosphate is binding to the iron, preventing the decomposition of the hydrogen peroxide.